All right, here we are. So I've said what I'm going to about gregarines, and by this stage, everybody in class, well, almost everybody will have seen some. We found quite a lot of them in our South American roaches. Gregarines are not as studied as much as other apicomplexans because A, many of them aren't particularly harmful and may not qualify as parasites, and B, they are monoxenous. They only parasitize one host, and it's always an invertebrate. They don't parasitize any critters of medical or veterinary importance. But this next group most certainly does. This is the coccidia. Quick review of the gregarine life cycle. This is one picture that you've got in your lab handout. If we start with stage 1, that is an infective sporozoite. That's the stage that's eaten, typically passes into the mouth, burrows partly into a gut cell, and transforms into stage 2. That's a full-blown trophozoite. Well, then 2 trophozoites decide that they like each other very much, so they make a special hug and form a gametocyst. That's stage 4. At least a few of you got to see gametocysts in lab. And within that gametocyst, each one divides many times to make gametes. And then the gametes pair up. And then we get to a mature gametocyst by stage 7. The gametes pair up to form zygotes, but that's the only diploid stage in the life cycle, and each zygote immediately divides by meiosis and again by mitosis to make 8 haploid sporozoites, each one in an oocyst. So you have a bunch of oocysts inside a gametocyst. The oocysts break out, and in some species it's kind of entertaining how they do that. There'll be these chains of oocysts that just kind of unroll out of a gametocyst. One of these days I'll have the patients to stare at cockroach poop down a dissecting scope until I can see that happen. This semester is not one of those times. And then the oocysts open up, the sporozoites emerge, and it's the circle of life. Coccidia change that in several ways. For one, they do parasitize vertebrates, including humans, and including animals of veterinary importance, livestock, fowl, chickens and ducks and all that, and pests. Many coccidia have more than one host at different stages of the life cycle, so they're heterogenous. They're not restricted to a single host like gregarines are. Sporozoites will also, coccidians, penetrate host cells completely. Gregarines, the ones that you've seen, I don't think do this at all. They'll penetrate with that epimerite kind of anchor on the front, but they don't burrow in completely. Apparently in some species they do burrow in completely, but it's a transient stage. It doesn't last. But coccidians can penetrate host cells completely, becoming fully intracellular parasites. Then the fourth big difference is that sporozoites undergo a stage called merigony, and that is repeated rounds of cell division when they're inside their host cells. The only real division that you saw in the life cycle of the gregarines was first division to make gametes, and then those zygotes divide to make 8 of those infective sporozoites. The trophozoites, the stages you saw yesterday or the week before, those don't divide. In coccidians, the equivalent stage does divide and will go through repeated rounds of cell division inside the host cells. That creates a multinucleate stage, a blob of cytoplasm with lots of nuclei called a schizont. And then the schizont divides into a whole bunch of a stage called a merozoite. That's a kind of swarm stage that bursts out and can infect more cells. So in the gregarines we receive, the trophozoites don't divide. In the gregarines, the trophozoites don't divide. In coccidians, the equivalent stage does divide, and you may have several rounds of asexual multiplication within the host. So the host of a coccidian may have a much greater parasite load than the host of a gregarine, just because the parasites can reproduce asexually and multiply exponentially for a time. They do resemble gregarines because some of those merozoites will transform into gametes that form oocysts, which are excreted in the feces. That's something you've seen. And within that oocyst, zygotes form, they divide by meiosis and create haploid sporozoites. And again, that's what you've seen in the gregarine life cycle. So stages 4 through 8 have pretty close counterparts in the coccidian lifestyle. The big difference is that stage 2 there, the trophozoite, can penetrate completely into an intestinal cell, and it can divide asexually in there. So there's a typical coccidian life cycle. This is Emeria. Different species of Emeria infect chickens, rabbits, dogs, cats. This is one of the ones of great veterinary importance, although I don't think people normally get this. If we start over at the right-hand side, you can see that the sporocysts release sporozoites. That's that little infected stage. And the sporozoites penetrate into gut cells. But once they're in the gut cell, they're in there completely, unlike gregarines, and they can divide repeatedly, asexually, forming a stage called the schizont. The schizont then divides into a bunch of merozoites, and they all go bursting out, and some of those merozoites can reinfect new cells. So one sporozoite getting introduced into the gut might produce dozens of merozoites, many of which can infect more cells and repeat the cycle, albeit in coccidians a limited number of times. That's something that you're going to see in spades when we talk about things like malaria. One of the symptoms of malaria is you get a burst of chills, fever, and the headache from hell every 3 or 4 days, depending on what species you've got. And that's because malaria undergoes the same kind of rounds of asexual reproduction within your blood cells and liver cells. They do the schizogony producing merozoites the same way that coccidians do. Anyway, some of those merozoites transform into gametes. They'll pair up, they'll form an oocyst, and the oocyst is the stage that's shed in feces. And the maturation of the oocyst requires oxygen, so these things have to mature outside the host body. So once they're out of the host body, the zygote inside an oocyst will divide into, well, first into 4 haploid cells, and then and then they'll divide again and form zygospores within the oocyst. And then that's the stage that gets swallowed by a new host. So similarities but differences with the gregarines that you've seen. Sorry, somebody went ding dong. Now I don't see who it was. Right. Ah, there. Somebody went ding dong. All right, this is a pop culture reference you may not get at your age, but every time this thing goes ding dong, I want to say, Avon calling. Okay, so there are 1,700 described species of Eimeria. They're very host specific, and you may have several species of Eimeria that can parasitize one host. Eimeria tenella is a severe pest of chickens, and here's what this looks like. There's a sporozoite penetrating into a host cell. You can see the nucleus of the host cell labeled N, and in the cytoplasm of the cell, you can see there's a little vacuole that has this banana-shaped object inside. That's the sporozoite. That's the equivalent of the gregarine stages that you were seeing in the gut. The difference, of course, being that this can penetrate all the way inside. Inside the host cell, the sporozoite rounds up and the nucleus divides repeatedly, and you end up with a multinucleate mass called a schizont. And then the schizont begins dividing its cytoplasm into separate cells. And after about 4 days, the host cell ruptures, and the little swarmer cells, the merozoites, burst out and start spreading. And the problem with Eimeria tenella, the chicken disease, is that this tends to happen in sync. So if you're a chicken, you're just sitting there minding your own business, and all of a sudden many thousands of your gut cells all burst open at once and release merozoites. And then 4 days later, millions of your gut cells all burst open at the same time and release millions more merozoites. And so you get repeated bouts of terrible cell damage leading to bloody diarrhea if you're a chicken, which is not fun. This is what this looks like. These are chicken ceca. That's blind pouches in the gut where fibrous material is fermented, analogous to your appendix or the ceca. There's lots of mammals that have a cecum, especially leaf-eating mammals like rabbits. On the bottom, you can see what an uninfected cecum looks like on the inside. Up at the top, you can see an infected cecum that's been cut open, and you can see that the lining is bloody and swollen, and I guess those are probably clots of blood inside. You can see the damage that poor chicken ceca are taking because of damage to the epithelium by Eimeria. And then there's a schizont. That's about ready to burst. That will release the little swarmer merozoites. This will repeat 2 more times, but it can't go on indefinitely. That means that coccidian infection is what we call self-limiting, meaning that if the host doesn't die, the infection will eventually burn itself out. This is not true of malaria, by the way, which can keep doing these rounds of schizogony for years. But it's true of coccidia. If your chicken can get through it, the infection will eventually come to an end because there's a limited number of times that those trophozoites can undergo schizogony and then burst out with the swarmers. Okay, and then once you're up at the 2nd or 3rd generation merozoites, they will penetrate new host cells and either turn into single macrogametes or little flagellated microgametes. That's a cross-section through chicken intestine. You can see a lot of ordinary cells, but if you see those little, white spaces with a kind of pear-shaped object inside, those are macrogametes. That's equivalent to the eggs of Eimeria. They hunker down in there. The microgametes will penetrate into host cells that contain macrogametes and form oocysts. The oocysts then form a thick wall, and the wall has a little pore where the sporozoites are going to come out. That pore is called a micropyle. And the oocysts, get sloughed off, leave the host cell, and they're passed out in the feces. When they're freshly passed, what they look like is stage A right there. They actually need to be in the presence of a couple of oxygen for a couple of days to finish the life cycle. So once they're outside the body, out in the nice fresh air, the oocysts first divide by meiosis to create 4 cells called sporoblasts, and you see that in the middle. That's stage B. And then the sporoblasts divide by mitosis to give 8 sporozoites. That's the infective stage. And that's what you're seeing over there on the right at C. Each oocyst contains 4 spores, and each spore contains 2 sporozoites. So that's said to be a 1-4-2 arrangement. And that's typical of genus Eimeria, this 1-4-2 pattern. So there's a mature Eimeria oocyst. Up at the top, that bright spot is the micropyle. That's that little pore that the sporozoites will break out of. You can see there's 4 sporocysts. They're shaped, I guess, a little bit like lemons. And each sporocyst contains 2 sporozoites snuggled up right next to each other. It may be kind of hard to see that there are 2 cells, but you should be able to see 2 nuclei in each sporocyst. So there's the 1-4-2. One oocyst contains 4 sporocysts, each contains 2 sporozoites. That matters because this is the way you identify genera. So for example, this is called isospora, and instead of a 1-4-2, it's got a 1-2-4 configuration. Each oocyst contains 2 sporocysts, and each sporocyst contains 4 sporozoites, and in this case, a little bit of leftover stuff called a residual body. This is the kind of thing that veterinarians have to look for. If you take your dog or your cat in and they do a fecal swab or a fecal sample, this is what they're looking for. The past couple of times I've taught this course, I've had students that have been interning in vet offices, and this is apparently pretty common. You look at a lot of poop if you're a vet, and seeing this sort of thing is pretty common. I think I was told Eimeria is a pretty major cause of severe diarrhea in puppies. It's one of the things that puppies can die from if they're not cared for properly. Okay, cyclospora, qu'est-ce que c'est? Never mind, old song, before your time. Cyclospora has small oocysts with a 1-2-2 configuration. Each oocyst contains 2 sporocysts, and each sporocyst contains 2 sporozoites. There's one species that does infect humans, and it doesn't seem to be that well known, but it was reported from North America in 1990, and it can cause outbreaks of diarrhea that are transmitted by our old friend, the fecal-oral route, consumption of food and water that's been contaminated with human feces. I don't believe it's a major threat to public health, but there are occasional outbreaks of cyclospora-induced diarrhea in people. Pre-meds might conceivably encounter this. Okay, Sarcocystis, a family of coccidians called Sarcocystidae, adds a new wrinkle because they're heterogeneous. Aimeria, the one that causes puppy diarrhea and chicken diarrhea, is monogenous, right? It's only got one host, except for that little time outside the body. It will spend all of its life cycle inside a chicken or puppy, or rabbit's another one that can get coccidia a lot. But coccidians in the family Sarcocystidae have two hosts instead of one. They're heterogeneous, and here you've got both hosts. This cat that is in the process of eating a mouse. The oocysts get eaten by intermediate hosts and form cysts in the intermediate host tissues, which in this case the intermediate host is the mouse. When the cat eats the mouse, the cat gets infected, and the cat serves as the definitive host for these coccidians in the Sarcocystidae. These are what we're going to be looking at in lab next week and the week after, because there are several species that are very common in livestock animals. We'll be working with one, by the way, that is not known to infect humans. Although this is one where we are going to want to take lots of sterile precautions. So I'm just letting you know in advance, this is not one that's supposed to be infected, but you're still going to want to be especially careful about washing up and cleaning everything. I'll need to have the lab in better shape too. And the most famous sarcocystid is toxoplasma. And this infects something like 50% of the human population. We are not normally the host. And there's no way we can pass it on except in very rare and unfortunate circumstances where humans get eaten by their own cats. If anything happens to me, I assume that's what my cats are going to do with me. I have no illusions about the fact that I have predators. The only reason they haven't killed and eaten me already is that I'm bigger. Anyway, never mind. But humans can get this through the fecal-oral route or through handling a lot of cat feces. And this is why my wife made me empty the litter box. She used to take care of it, and then when she got pregnant, I had to start doing it. Now that our son is almost 10, we might have to rethink this arrangement a little bit because I've been doing it for 10 years. But you can get it from contact with cat poo. Most people, it's not symptomatic, but it can be severe in immunocompromised patients. People with immunosuppression therapy for organ transplants, people with HIV infection, people in chemotherapy, things like that. Anytime your immune system is weakened, toxoplasma can be dangerous. And it can also cross into the placenta and cause birth defects, which is why my wife made me start emptying the litter box when we found out she was pregnant. So it infects cats when they eat infected rats, and the life cycle in the cat is just like what you've seen. The trophozoites will penetrate the intestinal cells. In there, they'll undergo that schizogony. So you see some labeled S right there over on the left. That's infected cat cells where the trophozoite has divided and is creating those swarmer cells. Okay, so the swarmer cells burst out, they infect more cells in the cat gut, and ultimately some of them form gametes, either macrogametes, labeled MA over there on the right, or microgametes, which are analogous to sperm. And the microgametes burst out, fertilize the macrogametes, make oocysts, and the oocysts pass out into cat feces. That's what we've already seen. That's what the oocysts look like. Cats are the definitive host because they're the ones where sexual reproduction happens. The only definitive host of toxoplasma seems to be members of the cat family. Domestic cats certainly, I don't know if they can infect all of the wild cat species. They can probably infect a lot of them. One of you was telling me she was thinking about a career as a vet working with exotic animals, so maybe you can tell me this eventually. But the intermediate hosts can be a very wide range. Livestock, humans, rodents, a lot of mammals can serve as intermediate hosts if they come into contact with toxoplasma oocysts. And when an intermediate host swallows oocysts, the oocysts release rapidly dividing cells called tachyzoites. And they're called tachyzoites because they wear these bright-colored Ed Hardy shirts with a lot of Axe body spray. Actually, I'm kidding. Tachy is Greek for fast, as in like a tachometer is the dial on your car that measures how fast your engine is moving, your tachometer, the tach, whatever, same root. The tachyzoites are the ones that live fast. Unfortunately, they don't also die young. It's not like James Dean here. The tachyzoites penetrate cells in intestinal connective tissue, lymph nodes. They can get into your liver. They can penetrate into white blood cells, the macrophages, and so be spread throughout the body. And these are the ones that can cross the placenta and infect a fetus. They will penetrate host cells, divide inside them, and burst out and spread to new cells. So the tachyzoites spread rapidly, they divide fast, they spread through the body. Once they've spread, they'll start transforming into a much more slowly dividing stage called a brachiozoite. Maybe that's bradyzoite. So-called because clearly all of the members of the Brady Bunch were infected with these. Actually, I'm kidding. Brady is Greek for slow. So the slow livers, brachiozoites, penetrate into host cells, surround themselves with a tough wall, and form tissue cysts or zoetocysts, which can last for years. This is the stage that, if you're an infected rat, the brachiozoites will form in your muscle tissue and just sit there, possibly for your entire life, until you get eaten by a cat, in which case the cat eats the zoetocysts, which burst open and release trophozoites, and here we go again. So that's what brachiozoites look like. There's a fully mature one over there on the right. This is the stage we're going to be looking for in grocery store beef in the lab we're going to do next week and the week after that. It's very common. It isn't particularly symptomatic for the cow itself, but it is one of the leading causes of spontaneous abortions in cattle, which is a problem if you're breeding the things. And it is not known to infect humans, but it is very widespread among livestock, and I've always been able to find these whenever I've gone looking. So that's what we get to do next week. We're going to be looking for these brachiozoites in beef. There's a mature zoetocyst that might contain hundreds of little brachiozoites. And then when infected tissue gets eaten by a cat, the brachiozoites burst out, They infect the cat's intestinal epithelium, and I won't sing it this time, but it is the circle of life, and it moves us all, and we take our place on the circle of life. The last thing I'll say is the really cool thing about toxoplasma, and not only toxoplasma, there's a lot of parasites that do this. There's a lot of parasites that engineer host behavior, that not only infect a host, they can make the host behave in such a way as to enhance the parasite's chance of surviving and reproducing, even if that behavior puts the host at a disadvantage. Infected mice and rats lose their fear of cats. In particular, what they lose their fear of is cat urine. I've been told that rats and mice instinctively exhibit terror when you let them sniff some cat urine. And someone told me once that this is actually used for testing anti-anxiety drugs. That, if you're testing an anti-anxiety drug, you dose a bunch of rats with it, have them sniff cat urine, and the ones that go, hey, whoa, man, is that cat urine? Dude, wow. Well, that drug works. It's used as a model system for studying anxiety. So uninfected mice will squeak and run like hell when they smell cat urine. Infected mice are attracted to it. Infected mice will just walk right up to the cat and go, hey, man, how's it going? And the cat, of course, eats the mouse, and then the cat gets infected. And it seems that this causes permanent change in the brain, because infected mice continue to have no fear of cat urine even after their immune systems had cleared the parasite to undetectable levels. So here, what you're looking at is, on the y-axis, aversion ratio is the ratio of time spent away from some urine to time spent close to it. The rats don't show any particular aversion. They spend about as much time close as away from uninfected rabbit urine, but you can see they spend a lot less time away from, okay, I see what we're getting at. The circles show what uninfected rats do. They don't bother with uninfected rabbit urine, but they freak out and stay away from uninfected bobcat urine. Okay, with me there. Type 1 and type 2 are the responses of rats that have been infected with 2 different strains of toxoplasma. And you can see for the triangles and the squares, there's no difference. They spend equal amounts of time close to and away from both bobcat and rabbit urine. Infected rats don't care about bobcat urine. And the patterns still persist 4 months after infection. By that time, the rats no longer have any parasites in them, but they're still lost their fear of bobcat urine. So the brain of the rat seems to be permanently changed by infection with toxoplasma. There are claims that toxoplasma can mess with human behavior as well. That people that are infected with it are likelier to engage in thrill-seeking behaviors. So if you know someone whose last words are likely to be, hold my beer and watch this, it is possibly due to toxoplasma infection. This is being looked at right now, that human behavioral tendencies can be altered by getting infected with toxoplasma. Sleep well, everybody. All right, I'm going to go ahead and stop the recording on one of these buttons. Got to be. Okay, here we go. And we are done with